Hello and welcome to today's special GeneWest Week webinar, Metagenomics for Biomarker Discovery, Applications in Precision Medicine. I am Dr. Sumit Kumar, Senior Product Manager at GeneWest, and I will be moderating today's webinar. I am very pleased to introduce today's presenter, Edward Messick. Edward Messick is a bioinformatics analyst at GeneWest. He completed his MS at the New Jersey Institute of Technology, where he studied bioinformatics, focusing on machine learning and human disease risk prediction based on genomics. With his years of study and 5 years of experience working in a hospital, fueled his passion for research in human health, disease, and drug development. Before joining GeneWiz, Edward worked in various roles within biotech and pharmaceutical companies, developing biologics for a small pharmaceutical company, as well as at Quintiles, managing clinical trials and clinical data. His major interests are in drug development, human genetics, and development of bioinformatics system to aid in research within these fields. If you have any questions for Edward during the webinar, please enter them into the Q&A section, and we will try to address as many as possible at the end of the session. With that, please welcome Edward.
Thanks for the introduction, Sumit. Today I'm excited to talk about metagenomics and its applications in precision medicine, specifically about how we can develop biomarkers using metagenomic techniques. First, I'll give an introduction to what metagenomics is, a brief history of the technologies involved in metagenomics, and then I'll talk about the broad applications and sources of data in this field. Metagenomics is defined as the study of all genetic material in a community of microbes. Likewise, it is known as ecological or group genomics. Right now, metagenomics is rapidly growing as sequencing technologies advance. Throughout this talk, I may refer to metagenomics as meta. The power of metagenomics is highlighted when we consider that 99.9% of microbes aren't cultivatable, enabling us to examine and analyze 10,000 times the microbes and genes present in a given environment compared to traditional wet lab techniques. Metagenomics also enables us to peer into all domains of life, from bacteria to archaea to viruses, fungi, or protozoa. So there are three major high-level approaches that can be used to summarize the goals of metagenomics. The sequence-driven approach is asking who is there or what microbes or genes are in the sample. The function-driven approach is asking what are the microbes doing and aims to study the genes and biological pathways present in the studied environment. And finally, we can perform comparative analyses between samples or groups. And this is where we're going to find good biomarkers. So as you can see here, there's a timeline of the technologies that have enabled us to do metagenomic analyses. We only have been able to easily study communities of microbes over the past 10 years or so as high throughput sequencing technologies have developed and become more cost effective. The term metagenomics was only termed in 1998 by Joe Handelsman when referring to an environmental metagenome. So if you look to the graph at the top right of the screen, you can see the exponentially growing trend of microbiome-related publications, which continues into 2017. It really is a booming field right now. Before I move on to the next slide, I'd like to just add one more point to the timeline, which is this webinar. So before I go into more about what techniques we can use to perform metagenomic analyses, I want to highlight a few of the many sources, industries, and applications of metagenomics. In this slide, you can see the non-exhaustive list of environmental sources for metagenomic samples. Anything non-sterile can be studied with metagenomics, from the very diverse and complex soil or ocean samples to the host-associated samples from humans or animals, to plants and even in space. Often studies will compare microbiomes between different sources. The applications of metagenomics are so broad that it can be useful across many entirely unrelated industries, from agriculture, where researchers can study plant health or use meta to improve crop yield, to drug discovery in the pharmaceutical industry, to monitoring the entire chain of food production. Metagenomics is a powerful way to study in depth the microbial world that may be directly affecting your industry. Metagenomics has powerful applications in epidemiology, where it can be employed to trace the source and spread of infections throughout the world, as well as in facilities like in hospitals. This slide is an extension of the previous slide, giving some more concrete examples of what can and has been achieved with metagenomics. There are many examples of metagenomics revealing novel genes, species, and a long list of enzymes that have been discovered. Many of these discoveries are coming from high-complexity microbiomes in soil or ocean samples. I'll go into more detail for the rest of the presentation about biomarker development. There have already been several drugs developed using metagenomics. If you look to the right of this slide, there's a table of antibiotics which were discovered through metagenomic screening. In the same paper, you can find the list of enzymes I mentioned. These examples have been established as powerful applications for metagenomics already. Right now, and in the immediate future, metagenomics is being used for pathogen detection, for developing companion diagnostics, and for cancer diagnostics. For example, recent studies have found signatures in the microbiome that can be used for the detection of colorectal cancer without need for invasive procedures. Our microbiome is also implicated in pharmacogenomics. Metagenomics is also useful in studying antibiotic resistance, and of course the complex interactions between microbes. Before moving into the nitty-gritty of the common methods employed in metagenomics and how to develop biomarkers using metagenomics, I want to highlight the booming microbiome market. The microbiome market is expected to grow to over $3 billion globally by 2025, with about $1 billion of that being the human microbiome market. I mentioned 23andMe here because the recent FDA approval, which allows them to disperse health and disease risk information to consumers directly, is enabling many startups to pop up in this area. You can see toward the bottom of the slide some of the recent startups in the microbiome market. To the top right, you can see that in the past year there has been a large investment from venture capitalists into this market. The reason I believe this is happening is because our NGS technologies have become more cost-effective and scalable, while the current boom of microbiome studies have revealed just how much the microbes around and within us are affecting things. So now I will go into the methods and techniques in metagenomics and then go into detail about how to screen for biomarkers. This slide is a very high-level overview of the wet lab side of things, where we are either doing amplicon sequencing or whole genome shotgun sequencing, both of which I'll get more into in the coming slides. So here you can see that there are two major categories for sequencing our samples and extracting genetic information. Amplicon sequencing, like targeted 16S sequencing, has been widely used over the past decade, but is becoming less relevant as shotgun sequencing becomes cheaper. However, 16S is still useful now, though much less powerful, as it can only be used for classifying bacteria and cannot directly provide a good profile of genes present in a sample. So for the rest of the talk, I'll mainly be focusing on shotgun sequencing and analyses attached to this, because it enables us to look at any domain of microbial life like bacteria, protozoa, or viruses, while also enabling us to do functional analysis of the genes and biological pathways present. Shotgun sequencing also has better resolution than 16S at the species level, often even achieving good specificity to the strain level. Typically, when we talk about metagenomics, we're talking about shotgun sequencing, but amplicon sequencing is sometimes referred to in this manner. As you can see, metagenomic analyses can be broken down into two major categories. Taxonomy, where we are defining what taxa are present in the sample, often called OTUs, or operational taxonomic units, like a species or a genus, and producing associated abundances of these OTUs. And then the second analysis would be functional analysis, where we are producing catalogs of the genes present in a sample, as well as the biological pathways associated with these genes. In this slide, I'll go over a typical workflow for doing taxonomic analysis. As you can see, there are a few ways to approach this analysis, but I'll go into what I think are the best current methods. When we look at the data typically used for shotgun metagenomics, we are using short reads produced by a sequencing platform like Illumina's MiSeq. So first, what we need to do is some form of quality control by filtering out low quality reads and trimming for adapter sequences. Then, if we're studying a host-associated metagenome, we need to align to that host genome and subtract the host reads so that we are only left with the reads from microbial genomes. Now, depending on what you're studying and other things like our computing power, we can approach classifying these reads taxonomically in a few ways. The top arrow uses marker gene analysis and is typically what we do if we have low computational power or we have a database built specifically for what we are studying. But I think this methodology is becoming less relevant as our computational power and NGS technologies grow. I believe the resolution here is weaker than comparing directly to a large database of genomes. The bottom arrow is another method which I would not highly recommend, which is assembling the shotgun reads. I wouldn't recommend this for taxonomy. The problem with this approach is that we are doing de novo assembly, not reference-based assembly, of a large number of short reads, which requires revision and often will lose much of our data, while also producing chimeric reads that need to be accounted for and removed. It can be very useful if you are looking for one or a few specific taxa, because in this way we can calculate the coverage of that taxa's genome. Now, if we look at the middle workflow, this is the best general workflow to follow for doing taxonomic classification. The binning step is attempting to classify reads to a given genome, and depending on how you are doing your alignment to a sequence database, it can be very computationally expensive. I use a Biomarker-based lowest common ancestor classification method, which is going to be very fast to align to the sequence database with excellent specificity. Really, it has a great balance of speed and accuracy. However, it requires loading the database into memory to achieve this, so you need proper computational power. You can also use methods that are based on pre-computed databases like a hidden Markov model database to do fast calculations, but there are limitations, and they often only contain a subset of the many genomes available. There are many ways to align that produce similar results as far as sensitivity, however. The best way, in my opinion, given proper computational resources, is to build a database of all known microbial genomes and to align to that database, returning the best match for each read in the database as its classification. Again, I think indexing your database by K-mer and aligning this way will produce very accurate results very quickly. In this slide, I'll go over how to do functional analysis, where we're trying to understand the genes present in the metagenome and then linking those genes to biological pathways or functions. By doing this from genetic data, we are defining the functional potential of a metagenome rather than the expression, which we look at with metatranscriptomics, another topic altogether. To the left is a basic high-level workflow showing that first we do gene prediction to produce a gene catalog or list of genes, and then we perform functional annotation against databases like KEGG, Eggnog, or Go, for example. To the right, you can see a more detailed workflow, including the typical QC step. Don't forget to remove host reads if need be. I've seen better results doing de novo assembly than direct alignment of reads to a database of known genes, so I'd recommend doing assembly for functional analysis, then revising your assembly, and don't forget to consider that this step will produce chimeric reads that we need to filter. So then we do the gene prediction by aligning to a database of known genes, and we cluster these genes into gene catalogs, which will then be annotated against databases like KegGo or Eggnog. There are many databases out there which are specific to one application and can often be better than a large or general database. After annotation, we do our downstream analysis, which you can see listed below, broken down into functional composition, where we can actually infer taxonomy here using marker genes, but again is not the preferred method. And then we can do comparative analyses between samples and their functional profiles, and that's where we're going to look for biomarkers. This slide is here to highlight the commonly used visualization and comparative analyses that we'll see in a typical metagenomic study. It can be important for a researcher to visualize the data to better understand or distinguish the statistical differences between metagenomes, but also to provide this information in a way scientists can easily comprehend. Heat maps, histograms, and stacked bar graphs are good visualizations for looking at the entire metagenome of an environment and to compare the metagenomes of samples to one another. Corona charts are useful in exploring an individual metagenome. Looking at networks, PCA plots, and other clustering plots, these plots can help reflect the differences between metagenomes that we'd find statistically and can aid in biomarker screening. Diversity measures like alpha diversity, or the diversity within a metagenome, or beta diversity, the differences in diversity between metagenomes, are measures drawn from metagenomes that can be biomarkers themselves. So I've been talking about them this whole time. What are biomarkers? Biomarkers are measurable indicators of some biological condition or state that can indicate some phenomena like an infection, disease, or environmental disturbance. There are a few classes of biomarkers I've listed here. Functional biomarkers are what we think of normally, or as a classical biomarker, which includes genes, proteins, or metabolites. Taxonomic biomarkers can be a specific species or OTU, like we'd often find in infectious disease states. A new class of biomarkers that has recently emerged are microbiome-based biomarkers. These microbiome-based biomarkers include the metagenome itself and some of the measures of that metagenome which I mentioned in previous slides, like alpha diversity. Going into a little more detail about these biomarkers, there are several types we can consider. For example, we can start by defining the full microbiome or profile of OTUs as a baseline. Then a potential biomarker could be a shift of the microbiome between conditions, environments, or time points. Disturbance of the ratios of microbes to one another in a microbiome can be indicative of a given condition. We are seeing diversity measures like alpha, beta, or gamma diversity producing biomarkers. For example, in recent studies we have found that the diversity of the vaginal microbiome can be a strong indicator of preterm birth. Primarily, we will be looking at differentially expressed genes, microbes, and pathways. This is where we're going to find strong biomarkers. So the power of metagenomics in this area is that we can really use it to develop any of these classes of biomarkers. Here's an overview of the high-level steps involved in biomarker discovery. The first step is designing your experiment and obtaining the samples and extracting the DNA. Here you will need to consider what you are trying to study and can develop some preliminary targets for biomarkers based on your biological knowledge. Are you looking for a functional biomarker? Then you have to do shotgun sequencing. The next step is where metagenomics is employed, in screening and selecting biomarkers statistically from your genetic information. Finally, in the lab, you'll need to validate your biomarker, which often needs to be designed specifically for your purpose, but there are some general methods we can follow. I'll mostly be focusing on the screening and selection of biomarkers. As you can see in the diagram, biomarker discovery is the first phase of many in the full development of a biomarker. Here I'll highlight some of the methods used for biomarker screening and selection, and over the next few slides go into more details about how to use these methods. So we've already identified the taxa and genes in our metagenomic samples. Now what we need to do is find the major statistical differences between samples of different conditions. We can use combinations of basic statistical methods like a t-test or Wilcoxon rank sum test, or we can do clustering by measures of unifrac distances with methods like principal coordinate analysis or principal component analysis to find separations in the data. Machine learning methods like random forest, linear regression, or SVM, support vector machine, can be powerful techniques to find features unique to a condition, which then can be explored in biological tests in the lab for validation. Some of the packages in R that can be used for differential expression are dseq2 and edgeR, which are very commonly used if you do this type of work. So when we are trying to statistically or mathematically find the differences between our samples, we need to first look at what our data entails. Do we have discrete or continuous variables? If we have non-value labels associated to our data, we'll need to do classifications of our samples, whereas if we have continuous numerical values associated to our data, we can explore predicting values to select biomarkers. Similarly, we can ask from machine learning approaches, do we have labels to our data which we can use in supervised learning to train, test, and revise our model by selecting features, or do we have to use unsupervised learning methods? In the next slide, I'll go into details of using one published complex statistical method for screening biomarkers called LEFSE. LEFSE is a published method for metagenomic biomarker discovery by way of class comparison, test of biological consistency, and effect size estimation. This addresses the challenge of finding organisms, genes, or pathways that consistently explain the differences between two or more microbial communities. So here's a high-level workflow of how this method works. Of course, you can see that metagenomic taxonomy and functional analysis has been performed. Then we're going to distinguish the differential features using LEFSE. The method employs a method called linear discriminant analysis for high-dimensional class comparisons, which is tuned perfectly for metagenomics, where often there are more potential biomarkers than samples themselves. LEFSE can determine the features like genes, species, functions, or biological pathways most likely to explain the differences between classes or conditions by coupling standard statistical significant tests with additional tests for biological consistency and relevance, as well as incorporating existing biological knowledge. Typically, class comparison methods predict biomarkers consisting of features that violate a null hypothesis stating there's no difference between classes. But this method also determines the significance of these variations. This approach was validated on human microbiomes, mouse models, and environmental samples, so it's very powerful for biomarker discovery. So as you can see, this method is layering statistical tests to determine the significance of the differential features between conditions or classes. Now I'll go into what this method entails. So the input data consists of a collection of M samples as columns, each consisting of N numerical features in rows. In this image, they are represented as a heat map depending on their value being high, medium, or low. Each class is the main condition to be compared in your study. So the first step analyzes these features, testing which values in classes are differentially distributed using a Kruskal-Wallis test. And then the features which which have a passing p-value, will be tested in the second step, where a Wilcoxon test will be used to determine features which violate the null hypothesis. So this is testing whether pairwise comparisons between these subclasses within each class significantly agree with the class-level trend or not. The features that make it through this layer of feature selection then are used to build an LDA, or linear discriminative analysis model, from which the relative difference among classes is used to rank the features. So after performing these statistical methods on our input data, we will have a final list of features which are going to be discriminative between classes or conditions, consistent within subclasses, and ranked according to the effect size with which they differentiate these classes. So at the top of this list, we're going to find great targets for biomarkers. Looking at this approach, we can see that it is important in the analysis of highly complex data like metagenomes to find not only the most differential features, but to make sure that these are statistically discriminative through layers of statistical tests. Here I'll briefly go over the approaches and considerations in validation. But like I mentioned, this is not done through a generalized bioinformatics method. Rather, a validation often needs to be designed specifically for each experiment and performed in the lab. So we've learned that biomarkers can be distinguished through statistical or machine learning approaches, and that we're looking for clear statistical discrimination between conditions to define a good biomarker. Or through machine learning approaches, we want clear clustering or classifications of our conditions. Again, validating a biomarker entails designing tests specific to your experiment, data, and type of biomarker. If you're looking to validate a functional biomarker, like a differentially expressed gene, for example, you can take approaches like designing primers and quantitatively measuring the presence of the biomarker using PCR, comparing between the two conditions. If you're looking to see if a specific taxa is causative in a disease, you may test in lab animals by introducing or removing that taxa. So there are some metrics for clinical applications that are generally good to look at, like defining an odds ratio or relative risk for a disease based on the presence of a given SNP. You can test the performance of a biomarker's ability to predict or diagnose with ROC curves or true positive rates or false positive rates, for example. I won't go into much more about validation, but as I have said, this is typically going to be designed specifically for your experiment. The goal in the end is the same, however, and it's to prove that your statistical link between a given biomarker and a condition exists to the degree that you previously detected in your screening phase. So lastly, I'm going to go into the applications of these biomarkers in precision medicine by discussing human microbiome-based biomarkers and some of the exciting research that has already been done in this area. The human microbiome is fascinating. We have over 100 trillion microbial cells that far exceed our human cells 10 to 1, as do their genes. We host all domains of microbial life in our body, and much like we have an actual fingerprint, we have a unique microbial signature. Right now, we still have a lot to learn in regard to how our microbiome interacts with our genetics, health, and development. It's interesting to note that we're born with a low abundance of microbes, but as we develop, we are mostly microbial. So we're born 99% human and we die 90% microbial. Before I get into some of the interesting applications of biomarkers in personalized medicine, I want to highlight the human gut microbiome because studies in this area are finding powerful links to diseases already. As you can see, our gut microbiome is linked to many other systems in our body and has been found to be associated with a laundry list of diseases, not all of which are gut disorders. For example, shifts in certain taxa will be indicative of obesity, as you can see in the table to the right. Everything we consume is modulated by our oral and gut microbiome. If you look to the lower right of the slide, there is a basic host model of biomarkers in environmental health that has been updated to include the gut microbiome as a barrier between what we intake and how we absorb, metabolize, and distribute substances. One powerful current application seeing clinical relevance is using microbiome transplants to cure C. difficile infections via fecal transplants from a healthy host. So as I mentioned earlier, our microbiome is implicated in many disease states one would not immediately imagine. For example, we are finding that cancerous tumors in many areas of our body have a unique microbiome signature that can be used for detection and potentially targeted by therapeutics. Biomarkers of disease states can be broken down into three major categories. Risk biomarkers that can be used to predict the risk of developing a disorder, diagnostic biomarkers that can confirm the presence of a disease, and prognostic biomarkers that can predict the outcome of a disease. Some of the strongest links between the human microbiome and disease can be seen in neurological disorders, metabolic disorders, cancers like in detecting colorectal cancer, in autoimmune disorders, and of course in gut-related disorders. If you haven't been monitoring this field, you'd be very surprised how many links we are finding to disease. Studies on this topic are coming out almost every day, whether it be in human or animal models. And if you look to the diagram on this slide, you can see some of these disease states that already have strongly linked microbial signatures, a few of which I've mentioned. I've separated the application of metagenomics in infectious disease from other disease states because one of the most powerful emerging applications for meta is in pathogen detection in a clinical setting. So we can use metagenomics to develop point-of-care assays which may be more powerful than our current diagnostics, but we can also use metagenomic sequencing and bioinformatics techniques as well to diagnose. Certain disease states have a high need for this, like Lyme disease, where there are numerous co-infections and poor diagnostic tests, or for meningitis or encephalitis, where over 60% of the cases are caused by an unknown pathogen. There are several reasons why we cannot detect these pathogens using current clinical techniques. And I'd like to highlight one lab at UCSF that's really pioneering the use of metagenomics in a clinical setting already for infectious disease diagnostics using SERPI. This will be the last slide I go over some of the applications we have for microbiome-based biomarkers. As I mentioned earlier, the FDA approval allowing 23andMe to disperse health and disease risk information is a milestone that is going to enable direct-to-consumer companies to perform genomic analyses. Similarly, we will be able to discuss the implications of our microbiome on health, lifestyle, and diseases. There's currently a large movement in genomics to start phenotyping, which will add a lot of power to the data we currently have. Part of the phenotype that needs to be considered is our microbiome, and part of phenotyping our microbiome is taking a baseline or healthy microbiome for each person to detect deviations from. There are many other applications for human microbiome biomarkers as well, including the ability to predict success in certain procedures like in bariatric surgery, which is something I've worked on recently. In this slide, I'd like to highlight a company called Ubiome, which is doing amazing work in studying the human gut microbiome and helping consumers gain insight into their health and lifestyle, while also providing clinicians a tool to diagnose gut disorders. So now that I've went over what metagenomics is, how to use metagenomics to screen for biomarkers, and how those biomarkers are relevant in medicine, I'll summarize the webinar quickly before taking questions. Metagenomics has broad applications in many industries, from studying the environment to food production to medicine. Microbiome science is a booming field, and studying the human microbiome will soon have significant impacts on health and disease. And as I've mentioned, these studies already are having an impact, as reflected by the growing market. Whole genome shotgun metagenomics is a powerful approach to studying the microbiome and really is the gold standard now that NGS technologies have become more cost effective. One must consider a large number of statistical or machine learning approaches for screening biomarkers using metagenomics. And finally, metagenomics can be used to develop biomarkers broadly and specifically in human medicine.
Thank you, Edward, for such an interesting webinar. It was very educational, and I'm sure listeners enjoyed the presentation and learned a lot about metagenomics and the bioinformatics workflows and how metagenomics can be used to detect and define biomarkers for some of the disease states. I see questions coming in, and we will try to answer as many as we can. So the first question I have on the screen is, and it has two parts. So the first part is, can whole genome shotgun metagenomics be applied to mixed populations of bacteria, fungi, viruses? And the second part of that question is there any type of organism that cannot be detected by this technique?
So that's really the power of shotgun metagenomics, is that you can study every domain of microbial life at one time. That it doesn't need to be separated into different analyses, and really the limitation on species is based on your database, and metagenomics can be used to find novel species. So in theory, you can find anything.
Great, that is very helpful. The second question which we actually have is that during your presentation you mentioned that this technology can be used for soil samples and to certain extent plant pathology as well. So the question is this technology or approach of metagenomics is limited by database targeting medically relevant organisms?
No. So there are many custom databases built specifically for one application, which may better serve that type of analyses. And one of those areas where there's a lot of databases is in agriculture and studying plant health.
And I remember you actually mentioned during your presentation that it probably would be better to actually use agriculture-based databases,
Yes.
rather than a very general database, which will probably actually provide much more and useful information.
Often it could be better and much less computationally expensive as well.
Okay, great. Thank you very much. And just for the interest of time, we will be taking one more last question. And again, there's two parts for this question. One is, does similarity of sequences limit the ability to distinguish species? And are there any known biases to the shotgun method versus the targeted approach?
So the targeted approach is going to be isn't going to be as good to distinguish species because you're using one part of their genome rather than the entire genome, where you're going to find more differences. But there is a problem with the databases in that often species have very similar genomes. So that's where you can do binning and you can do some curation of your database to make sure that you can clearly distinguish species.
Great. Thank you very much, Edward. And I would also like to thank our listeners for joining us and for their time. And I would also like to take this opportunity to remind our listeners that you can check other webinars we are hosting at genewizweek.com. And I would also like to invite you to join us again for our celebration of genomics as well as genomics researchers. So with that, I would like to thank you and have a good day.